Hello everyone, my name is Joseph Gaulin and today I'll be giving my senior seminar presentation. So I know a lot of females out there might be thinking or wondering, does your man or significant other of the opposite sex actually have a negative impact on your health? You hear a lot of people joke about it, but recent evidence suggests that it might be true in some rodent species. So today I'll be presenting to you on the effects of male presence and mating on female health and physiology. So first, I want to introduce you guys to the idea of life history trade-offs. So the definition is right here. You guys probably know what trade-offs are in general. It's when an increase in one thing results in a decrease in another. And what I'm referring to is trade-offs with regard to life history traits, which include lifespan, fecundity, which the definition is in the context of what we'll be talking about, referring to population biology. It's the same as fertility or the ability to produce offspring. And this concept of life history trade-offs is based on limited resources. So organisms don't have an unlimited energy pool to pull from, so they have to allocate different resources in order to maximize their fitness. Some examples are increased early life reproduction results in decreased late life reproduction, and increased overall reproduction actually decreases lifespan. So females in mammalian species have been known to have a very high level of parental care in their offspring. So the most obvious reproduction costs are of pregnancy or gestation and lactation. So the energy transfer being from the mother to the fetus through the placenta, or the mother to the neonate through the mammary glands, the milk production. So in many previous studies, due to the study designs, the effects of mating and male presence on female life history are actually masked by the effects of pregnancy and lactation, and this is because in a natural pregnancy, you cannot have that without a mating event or male presence. So previous studies, though, actually suggest a potential role for male sex cues, pheromones, and mating in female physiology and life history. So a couple different organisms, Drosophila, the fruit fly, they found that sexual anticipation, or the anticipation of the offspring event, is actually responsible partly for some of the effects seen in females. In C. elegans, roundworm species, it was found that males actually secrete compounds that decrease female lifespan. And then in the common house mouse, it's been shown that they can actually modulate their estrus cycling, which is the period when they can actually become pregnant. And they can actually maintain or abort pregnancies based on different signals they're getting from the males surrounding them, whether they're familiar or unfamiliar. So this leads to our overarching question: what are the effects of early life mating and male presence on female lifespan and late life fecundity in mice independent of pregnancy and lactation? So the rationale or reason that they're doing this study is because they want to reveal the mechanisms underlying how different life course factors or how people live their lives affect health and physiology, so that they can better understand the sex-based differences, so that they can better design studies and eventually relate this research to humans one day farther down the line. So this brings me to the focal paper that I'll be focusing on mostly in this presentation. It's this paper here, published by Garrett, Try, Smiley, Grattan, and Brooks in 2020. And it was published in this journal, the Proceedings of the National Academy of Sciences of the United States of America, and the affiliations of the authors: University of Otago, the Center for Neuroendocrinology and Biomedical Science Department, and then UNSW Sydney in Australia, the Evolution and Ecology Research Center. So first, I want to talk about the study design and methodology because it is a little complex. So for early life social conditions, I'm referring to 28 to 300 days of age. And in this period, male gonadal status was manipulated and we're studying female mice here. So here are the housing partners for those female mice. We have one of them in all 4 treatment groups being a female sibling, and this third mouse in the cage is what varies between the groups. The top one, we have an unfamiliar female. The next one, we have a castrated male, which show greatly reduced mating behavior and androgen-dependent sex signals, so they're not physiologically males. Then we have vasectomized males who do not produce sperm in their ejaculatory fluids, so cannot induce pregnancy, but they do still mate with females and have normal androgen levels. And then we have intact males who were sham vasectomized, so that just controls for everything except the actual vasectomy. And these males did make these females pregnant during this time span in this last group. Then at 300 days, the housing partners were removed, and all the mice from the early social condition groups were separated into one of 2 different late life cohorts from 300 days of age to death. The first one is called the non-reproductive cohort. This is when they were subsequently housed with just their female sibling, and this was used to assess lifespan. And the second cohort was the reproductive cohort, and this is when the female mice were housed with their female sibling plus an intact male who would obviously induce pregnancy in the female mice. And the mice in these cohort were used to study fecundity. So first, the researchers wanted to know, how does social condition affect body weight in early life? So our graph here, we have age on the x-axis throughout early life up to 6 months, body weight on the y-axis, and we have our 4 different groups of females here. Control in this graph would be the black line, so that's females housed with unfamiliar females in early life. So it was found that in both females housed with intact males in early life and females housed with vasectomized males, that those females had significantly higher body weight across all the different time points, 2, 3, 4, and 6 months, versus females housed with unfamiliar females. And this effect was not seen in females housed with castrated males. So this is expected in the females housed with intact males, because they are getting impregnated by the males, and obviously pregnancy is associated with weight gain. But the fact that we saw the effects in the females housed with vasectomized males led us to the conclusion, the takeaway, that androgen-dependent cues and or mating is what is leading, or is contributing to some of the effects seen in relation to increased body weight. So next, the researchers wanted to know, are these body weight trends from early life social condition groups maintained in both of the late life cohorts that I previously talked about? So first, we have the reproductive late life cohort. So these were the females that were housed with an female sibling plus an intact male. So in this late life cohort, it was found that yes, over the course of late life, the females housed with intact males and the females housed with vasectomized males had significantly higher body weights across this time period versus the females housed with unfamiliar females, the black line. However, in the non-reproductive late life cohort, it was found that only at the initial time point, so 12 months, there was the significant difference, the increases in both the females housed with vasectomized males and intact males, but this effect diminished and was no longer significant at any of the time points following the 12-month period. So no, not in that one. Takeaway, increased body weight that we saw in the early life social condition groups was only sustained in the reproductive late life condition when they were housed with an intact male. So next, researchers wanted to know, how does early life social condition affect subsequent late life fecundity? So just to bring you back to the experimental methodology, we're talking about late life now, because we're looking at late life fecundity. So the females that went through the early life condition and were paired with an intact male and their female sibling in late life, that's who we're looking at in this question here. So first we have a graph looking at days since pairing with the intact male. So on this graph, the 0 point on the x-axis here is actually day 300 because we're talking about only the late life. And then on the y-axis, you have cumulative late life offspring weaned. So how many offspring they weaned up to the certain point, the time point on the x-axis. So it was found that from 100 to 200 days after pairing with the intact male, that the females that were housed with vasectomized males in early life, that's the females represented by this red line, had more cumulative offspring from these times, but it diminished and was no longer effective from 200 days to the end of life. And then in the females housed with intact males, it was shown that starting at day 200 after initiation with intact male, they produced or weaned cumulatively less offspring compared to the control black line group. And once again, no significant difference in the females housed with castrated males in early life and the control group. So next, still looking at the same question, late life fecundity, we have fertility survivorship curves. So here we have, again, day 300 is the 0 point because we're looking at late life. And it was found time on the x-axis, and then the y-axis is the percent of females in each group that remained fertile, so still having reproductive events. So each point on the graph here in each of the lines represents one female in that group's last reproductive event. And it was found that females housed with intact males in early life had an earlier average age at last reproduction compared to the control, the black females housed with unfamiliar females in early life. So this was done using a log rank test, which is the common test used to compare curves like this. And also, we have another graph looking at the same question, how early life social condition affects subsequent fecundity. And we have litter number now. This is a little different. We have time on the x-axis here, days since pairing. So on the y-axis now, your 0 point is day 300. And then on the x-axis, we have litter number. So we're looking at how long it took to reach each sequential litter number. So basically, we're looking at the rate of litter production in this graph. And it was found that females housed with vasectomized males in early life produced their first 2 to 4 litters. So here, faster than the control females, which were the ones housed with unfamiliar females in early life. But this effect diminished after 4 and was no longer significant. So, all in all, takeaway, how does early life social condition affect subsequent fecundity? This study found that early life mating without fertilization increases subsequent fertility or fecundity initially upon pairing with the intact male. Also, we found that increased early life reproduction is associated with earlier reproductive senescence, and senescence is another word for lack of, so no more reproduction basically. Next, researchers wanted to know the other part of the main Q that I talked about. How does early life social condition affect lifespan? So now back to our study design. We're looking at the females that part of each of these 4 groups that were later housed with their female sibling only. That's what the cohort we're using here. So these are lifespan survivorship curves. So your more typical survivorship curve, quote unquote. Time on the x-axis here, it's actually showing all of life. So 0 to 300, this is where the early life social condition occurred. And then post day 300 would be the late life. And then we have percent survival on the y-axis. So each point here on each of the curves is representing the death of one female in that group. So a log rank test, again used to compare curves, found that the mean lifespan for females housed with vasectomized males in early life was about 84 days on average less than the mean lifespan of females housed with unfamiliar females in early life. So, takeaway, early life mating and or male presence has a deleterious or negative effect on survival in female mice. So next, researchers wanted to know if early life mating and or male presence is associated with decreased plasma prolactin levels in late life. So prolactin is a hormone secreted by the pituitary gland, and it's associated with pup care in mice, so maternal behavior and lactation. There is something called pseudopregnancy, and this is when a psychological or physiological sexual stimulus, so a mating event or the presence of a male secreting androgen pheromones, actually induces a neuroendocrine response that is similar to what you see in early females in early pregnancy. So this is characteristics of this is diurnal or daily surges in prolactin secretion and circulating prolactin in the blood. This actually leads to there's a negative feedback mechanism that reduces the amount of prolactin being secreted in reproductively experienced mammals because of the increased sensitivity to the hormone because it's been secreted so much previously. So that leads to the decreased circulating levels because less amount of the hormone is needed to induce that same response. So basically the researchers want to know, are we seeing this same neuroendocrine response in the females that are mating but not having fertilization or offspring production? So here is the data from that study. We have two different time points here. We have 18 months, 24 months, both during the late life period. On the x-axis, we have the late life reproductive status, so non-reproductive or reproductive, because all females in the study were looked at for prolactin. And then we have prolactin concentration on the y-axis. So it was found that females housed with vasectomized males in early life did in fact have lower plasma concentrations of prolactin in late life at these two time points, and this effect was actually additive. So answer, yes, only for females who mated in the absence of fertilization though, not in females that were housed with intact males. And it was proposed that this is most likely due to the fact that pseudopregnancy only lasts about 12 days versus 21 days for a gestational period, and this would most likely mean more mating events in females housed with vasectomized males because the females are not mating when they are experiencing pseudopregnancy or pregnancy. So in summary, the mating and sex signals contribute to life history trade-offs, so the effects on lifespan and fecundity that are usually associated with pregnancy and lactation. These are androgen dependent, as exemplified by the fact we didn't see any of the effects in the females housed with castrated males, only vasectomized males. And just to summarize the different effects we saw, increased body weight in females housed with vasectomized males and intact males. This was only maintained in the reproductive late life cohort of females. We saw decreased lifespan in females housed with vasectomized males in early life, about 84 days, which is modest. We saw increased late life fecundity initially in females housed with vasectomized males in early life, and then we saw decreased circulating plasma prolactin in females housed with vasectomized males in early life, and we saw that this effect was additive. So future studies are going to look into the mechanisms underlying these effects previously discussed. One will look into olfactory sensory systems, so that's olfactory, the smell in the nose here. Others will look into the effects of seminal fluid proteins, so that is proteins in the fluid secreted by the seminal vesicle, which is part of the ejaculatory fluid in males, and also different stress hormone pathways and growth hormone pathways that have been implicated in certain life history traits like lifespan and fecundity. So that is my presentation. I hope you guys learned something new, and I hope you enjoyed it.